In one of the earlier video, we have discussed about the UPR in brief. In that video, we have discussed that it majorly majorly gives activate three signals. One is IREA1 alpha, another one is PERK, and third one is ATF6. I have misrepresented ATF6 as ATF4 in the last video. So now I am correcting it that it is not ATF4, it is ATF6 that gets activated when the cell experience unfolded protein response. I will discuss about the other two parts separately. In this video I will discuss slightly in detail what is happening when IRE1 alpha signal gets activated. So before going on this, let us just briefly revise why UPR gets activated. UPR is unfolded protein response. This is from the name itself we can guess that this kind of response activated when the proteins does not get their native structure and there is accumulation of unfolded proteins beyond the normal limit. And most of this thing and many of the correct folding of the proteins occurs in endoplasmic reticulum. So this UPR is mainly occurred in the endoplasmic reticulum, and when there is a accumulation of unfolded proteins more than its normal limit, then there will be a stress generated inside the endoplasmic reticulum that will lead to dimerization of this IRE1 alpha. Usually it is present as a monomer. However, when the ER stress is exerted, then dimerization of this IRE1 alpha happened, and this lead to activation of further signals that was main aim is just to reduce this ER stress and make it back to its normal state. So the best way to do is that try to reduce the influx of the proteins inside the ER, and at the same time try to increase the amount of proteins that are responsible for correct protein folding. And the third way in which this stress can be reduced is to increase the flux of the degradation pathway, that is ERAD. In this case, when the stress is exerted in ER, ERAD is known as endoplasmic reticulum associated degradation. So when in order to reduce the stress, what the cell will do is that first it will reduce the influx of the protein inside the ER, at the same time increase the amount of unfolded proteins, influx of protein to ER reduced. And second thing that it will do is try to increase the amount of proteins that are responsible for protein folding, and we usually call them as chaperones. So increase the amount of chaperones. Chaperone should increase, and at the same time it should also increase the rate of degradation from protein degradation from ER. That is called as ERAD mechanism. So in these three things, cells will behave in order to reduce the ER stress. So when the dimerization occurs, first one of the signals that get activated through this dimerization is RIDD. It is known as regulated IRE1. What is this? It is known as Yeah, it is known as the regulated IRE1 degradation decay. Regulated IRE1 degradation decay. So what this will do is that at this moment the proteins that are general proteins that are not essential for the cell survival, it will this signal will reduce the translation of those proteins. So in this way it is do what this step is doing is trying to reduce the influx of the protein towards endoplasmic reticulum. So in this RIDD, the proteins that are not essential at this moment for cell survival, and those proteins will come in, and these proteins will come into the endoplasmic reticulum. So these two criteria need to be fulfilled. These two criteria need to be fulfilled by a protein, and if this criteria is fulfilled by proteins, then they might be downregulated in this RIDD pathway. And the next step, next signaling, it acts in two ways. One of the way is RIDD, and next way is XPB, XP1B. I think it's XP1B, XBP1. It is XBP1. Okay, so what usually this XBP1 mRNA is exists as unspliced. You can consider this as you can consider it as intron. So mRNA has two parts. One is intron, and other part is exons. And at this moment, what we know that exons are responsible for coding of the proteins, and what is the function of introns is not exactly known. So usually what happens in the process is that after this mRNA is produced, the introns get removed, and these exons connect itself through some biological process, and then it will translate. It will get translated into the protein. However, in case of XPB1, generally this intron does not get removed. When the stress is exerted and this dimerization occurs, then this helps in removal of this intron and removal of introns, and when this spliced XPB1 is called as XPB1S. What it will does is it will now go to the nucleus and upregulate the proteins that are responsible for this ERAD. This ERAD pathway. So what it will does is it will increase the flux towards degradation. So because the unfolded proteins are more in the endoplasmic reticulum. At the same time, what it will does is it will increase the amount of chaperones also. It will increase the amount of proteins. It will increase the amount of chaperones, or translation rate of the proteins that are chaperones will get increased. So this acts in like three ways, this signal. First is reduce the influx of the proteins through RIDD, and this XPB1 will increase the amount of ERAD proteins and chaperones. So this is one of the signal that attempts to reduce the ER stress and try to make it back to its normal position. That's all for this video. We'll discuss the other two signaling in the subsequent videos. Okay, thank you.